Bismillahirrahmanirrahim. Today I would like to share the knowledge about how any PhD scholar can defend the final thesis of the PhD. Similarly, my topic was evaluation of indigenous ELISA kit based on recombinant surface antigen 1 for diagnosis and surveillance of toxoplasmosis in humans and halal animals. And my name is Sarfraz Rahman. This is my 2018 PhD 2005. That is the registration number. And my department is the parasitology. Here is my supervisory committee. And next is the toxoplasmosis. So the Toxoplasma gondii, an obligate intracellular parasite, capability to infect the food animals, and approximately 1/3 of the world's human population is infected with the Toxoplasma gondii. And this one is the zoonotic parasite, and with the public health and animal health importance, and spread through the feces of the cats and through the eating of undercooked meat or raw meat. And in pregnant women it causes the serious problems: transplacental transmission from mother to fetus, miscarriage, permanent neurological damage, premature birth, visual disorders, and prevalent in Pakistan. Here is the data of the prevalence of the toxoplasmosis in Pakistan. So next one is the life cycle of the Toxoplasma gondii, and this is the life cycle of Toxoplasma gondii. These are the three infective stages: sporozoids, tachyzoids, bradyzoid. So the cat is only the definitive host for Toxoplasma gondii. Toxoplasma gondii has indirect life cycle and involves some intermediate hosts like the mammals and humans to complete its life cycle. This one is the life cycle. If I would like to describe this life cycle, so I would like to start from this cat, which is actually the final host. And this cat excrete the feces, and these feces containing the fecal ovocyst. And this fecal ovocyst that is spread the infection with the help of these four pathways. One pathway is that the rats or the rodents or the birds, when get these fecal ovocyst, and these fecal ovocyst changes into the tissue cyst, and this tissue cyst containing rodents, when it is eaten by the cats, and these tissue cysts enter into the cat again, and causes the infection of the Toxoplasma gondii. While similarly the other three pathway, one is that the cow and sheep that even eat that fecal ovocyst through during grazing or during drinking or any other pathway. So when they eat the get the fecal ovocyst, actually these fecal ovocyst changes into the that is the tissue cyst in their muscles, and when they are slaughtered and human beings eat these meat in the form of undercooked or raw meat, and these tissue cysts enter into the women or any other animal, other human being, and it will causes the infection of the Toxoplasma gondii. Similarly, the human being, if they eat contaminated water or vegetable with these fecal ovocysts, definitely they get the infection of the Toxoplasma gondii. But the fourth one pathway is that the cat, these cat containing the fecal ovocyst, and actually they get especially in the pregnant women, who carry the cats, and they definitely get the fecal oocyst through different ways, and then these women or the pregnant women get the infection, and this infection definitely causes the different problems in the women. So the next pathway is the different pathway, which is the blood transfusion method, in which the contaminated blood, when it is transfused to any other the man or women, then then the women and men get the infection. So this infection last that the pathway is the dangerous pathway, and it is the typical, or it is the most important pathway, because the pregnant women, if she gets the infection of the Toxoplasma gondii, and the infection goes or enter into the through the vertical transmission method into the fetus, and it causes the problem in the fetus, like microcephaly, macrocephaly, or hydrocephaly, or other different problems. So it is very important. So Toxoplasma gondii is a zoonotic parasite, and it is very important that it should be important that to get the awareness among the people, or men, or the women in the society, so they get or they cannot they cannot get the infection of the Toxoplasma gondii. So the SAG1 of the Toxoplasma gondii, SAG1 actually the surface antigen of Toxoplasma gondii, and it is also called the P30 and major antigen, a component of the Toxoplasma gondii. Good candidate for serodiagnosis of toxoplasmosis using the cloned gene of the Toxoplasma gondii, or SAG1, which is actually the recombinant surface antigen 1 protein, and extensively used based on the excellent immunogenic potential, and widely explored antigen for the detection of antibodies against the Toxoplasma gondii. So the statement of problem, and keeping in view the zoonotic potential of the toxoplasmosis, the meat of the food animals is considered the major source of its transmission to humans. The animals which are positive for toxoplasmosis are allowed for slaughtering in the slaughterhouses for human consumption, as currently no such testing is employed. So the developing an indigenous ELISA kit of toxoplasmosis to screen the animal is a need to identify the seropositive food animals. Similarly, no indigenous ELISA kit to screen the seropositivity of human being exists in the Pakistan. So this study was done, and this has been made the objectives, and the cloning of SAG1 in prokaryotic expression system to optimize the diagnostic value of rSAG1 through humans, sheep, and cattle, and to screen a sero of human, sheep, and cattle for toxoplasmosis. So these are the three main objectives, and this one is the methodology and results. This is the outlines or the flow. These are the main flow charts points. Actually, these are the main 7 points. The competent cell formation, transformation of PAT28 alpha SAG1, induction and expression of rSAG1, characterization of rSAG1, protein quantification, screening of sera through latex technique, and ELISA. So these are the 7. Next is the cloning of SAG1 in PAT28 alpha. The SAG1 was cloned into the PAT28 alpha vector at MCS between SHO1 and NHE1. PAT28 alpha was used as an expression vector. Novagen, the PAT28 alpha vector has kanamycin resistance portion, which is used as a selection marker. restriction enzyme that have a recognition site within the multiple cloning site are commonly used, and the gene of interest is most commonly subcloned into an expression vector for improved protein expression. So this is the map of the PET28 alpha. So next is these are the results, the conformation of transformed cell, and you can see that this is the transformed colony. It is the creamy or circular, and these are the two bands which indicating the that the gene of interest, and that is the vector. Here has two bands after double digestion with NHE and SH1 957 base pair and 5369 base pair. So the characterization of recombinant surface antigen 1 protein, this was induced by using 7.1 milligram with the rate of 0.5 to 1 millimolar IPTG in culture 60 ml, and then expressed RSAG1 was confirmed on 12% polyacrylamide gel stained with Comessi blue. RSAG1 was prominent at 35 kilodalton. This is the protein band, and you can see in this picture. So next is the screening of human sera through latex technique, and the results of LAT. These are the negative sample, these are the positive sample, these are the total 400 sample. So this is the latex slide, and you can see in the slide that is the agglutination which is present in the positive sample, and this is the positive control, and this one is the negative control. Human sera were evaluated by latex agglutination test. Out of 400 sera, that the 278 sera were positive by latex agglutination test. So the screening of human sera through commercial Toxoplasma gondii IgG ELISA kit, the samples were selected based on positive and negative sera, LAT and indigenous ELISA, and this is the result, and you can see in this table. So sensitivity and specificity of ELISA. Sensitivity and of the human toxo IgG ELISA kit was 98.5, while the human toxo IgG ELISA kit has had the 100% specificity, and you can see in this dot diagram. And next is the ROC receiving operating characteristics score that indicate the diagnostic efficiency, and the area under curve, that is the one which is the perfect test, and cutoff value is 0.398, and it was calculated by the ROC curve. And the outcome of the human indigenous ELISA kit, the 3 times economical kit as compared to the commercial ELISA kit, easily available within the short time, diagnostic lab, hospitals, different companies, made up of RSAG, one of the Toxoplasma gondii, more specific and more sensitive than the commercial ELISA kit, 100% specificity and 198.5% sensitivity. Cutoff value is equal to 0.398 to identify the positive and negative human sera. Timely diagnosis of the toxoplasmosis in pregnant women. So this optimization of sheep and cattle ELISA kit, you can see that the cutoff value for sheep 0.505, and the protein level for 0.125 microgram per ml. This is the 1:1600, while in cattle that 0.431 and 0.125 microgram per ml, and dilution is 1:100. And the screening of animal before slaughtering at the slaughterhouses. Our indigenous ELISA kits will be helpful for a diagnosis of the toxoplasmosis, especially sheep and cattle. So this is the brochure for awareness of the toxoplasmosis, and the brochure for awareness of toxoplasmosis was distributed among pet owners at University of Veterinary and Animal Science Law and different hospitals, and it was published in Aajkal Daily newspaper at on the 20th November 2020. So this is the first published paper. This one is the patent, and this patent for the ELISA kit has also been submitted. So next is this is the first published paper, and this one is the second published paper. This is a pictorial view of the lab work. Then this is the acknowledgement. We are thankful to HEC Islamabad, Pakistan Livestock and Dairy Development Department, Punjab, University of Veterinary and Animal Sciences, Lahore, my family, my parents, wife and care supervisor, all respectable teachers and all postgraduate colleagues. Okay, thank you so much.